I'm going to be speaking to you about productionizing molecular machine learning pipelines. Before we really get into the nitty-gritty of what this entails, especially since this is a space that is a little further from the usual machine learning applications, I thought it might be good just to provide a little bit of context. Um, One of the biggest drivers for molecular machine learning really is therapeutic discovery. And for folks who've been working in kind of this field, one recurring kind of phenomena is something called Eroom's law. It's sort of the opposite of Moore's law, which is that it seems to get about twice as expensive to design a new therapeutic every few years. We've, of course, with the COVID pandemic, seen how challenging it can be to create novel treatments, even with immense amounts of funding and the world's scientists focused on the effort. And part of the reason here is that just designing a new therapeutic is a very long and complicated process. I'm not really going to dig into a lot of the biology and chemistry here, but there's a lot of different steps involved, much of which really has more akin to scientific discovery than engineering. You have to say, form a hypothesis for what's the root cause of the disease you're trying to treat. You have to find a candidate molecule. You have to verify that this molecule is, not poisonous, can be ingested by humans in doses that treat the disease while not inducing bad side effects. You have to test in animals first before you give it to humans. You have to be able to manufacture it at bulk scale. And then finally, you have to be willing to give it to people. And again, kind of coming back to COVID as an example, vaccines are a little different than therapeutics, but a lot of the same difficulties arise there. The problems of testing, the problems of design, the problems of scaling up production. These are all kind of common throughout the therapeutic design industry and the pharmaceutical industry broadly. I think another kind of aspect of this industry that might not be as familiar to folks in the machine learning community is that proprietary techniques are still quite standard in traditional drug design. A lot of things are protected by core patents. There are some software vendors out there, but they typically retain very tight control of their software packages. You have things like per user site license fees that can be quite prohibitive. A lot of this technology is not easily accessible to students or hobbyists or new entrepreneurs. So there's a lot more barriers to getting involved in this space, which make it harder for newcomers really to explore interesting ideas. But I think in the last decade or so, there's begun to be a shift in the molecular design industry broadly, encompassing, of course, therapeutics design, but also fields like agricultural design and adjacent things like materials design. And in this case, it's what I'd call software-driven drug discovery, really software-driven molecular design. And the kind of key kind of driver here is that increasingly software-driven techniques have become increasingly powerful. Traditionally, the process of designing a new molecule was one where you'd have a lot of chemists, back in the old days, perhaps around a whiteboard, drawing out molecules and then testing them. In recent days, perhaps, looking at PowerPoint presentations and Excel slides, but still the core intellectual design done by human chemists. But increasingly, what we found is that software-driven techniques are increasingly helpful for human chemists as they undergo the process of design. The use of machine learning in particular has enabled systematic use of historic data to be able to design new molecules. This is kind of a very new field. A lot of these techniques are still primarily used in research labs and are only now really beginning to shift into practitioner hands. So this entire field, I would say, is still kind of in that stage where there's a lot of cool research results, but practitioners are just now really beginning to apply these techniques systematically in production. So another kind of thing that's been shifting in the last few years is that we know, of course, that modern software is really driven by open source projects. You have, large, widely used machine learning projects, things like, TensorFlow or PyTorch. You have, front-end packages like React, back-end things like Postgres, and of course, Linux underpinning the entire stack. So as drug discovery becomes increasingly software-driven, we are also seeing some penetration of more open source ideas. In particular, I'd say that the DeepChem project has been trying to drive a culture of open source development and open source software into the molecular design community. So DeepChem's goals really are to provide high quality scientific deep learning tools. It's an open source project that was originally founded at Stanford back when I was doing my PhD. But over time, we've grown the community into something that is, more open source, more anyone can join in. It's not purely an academic effort, and we welcome practitioners and researchers both to make use of the project and its tools. We have a whole collection of different tutorials. You can run them out on Colab. Uh, and yeah, here's something like 20 something tutorials at latest count, with more in the works. A lot of the challenges I think for entering this field are that it's been historically at this intersection of multiple hard disciplines. You have to know something of chemistry, a little bit of biology. You have to be a pretty decent software developer. And the goal of DeepChem and kind of its surrounding community is really to help lower these barriers by creating open, free tools. It's MIT licensed, very accessible to both companies and researchers, and providing a large collection of just tutorials and documentation, which we work to continually improve and make more accessible to folks. So with that kind of, I'll say a little bit about uh, the journey, I think, from research to production. So DeepChem itself is an open source project, started as like a pure research effort. The goal really was to enable duplication of existing research papers, and also uh, enable kind of broader use amongst the research community of these novel techniques. But as the field has matured, uh, the community has also begun to shift along the path, moving from a more research-driven field to something that's more ready to use in production. So in the rest of this talk, I'll give a kind of introduction to some of the scientific principles underlying molecular machine learning, and I'll also discuss some of the challenges that face the practitioner trying to apply molecular machine learning in real-world settings, and give a few tips and tricks from my experience on how to avoid or perhaps improve upon some of the challenges that we face. So just to start out, the first question is, what is molecular machine learning? Most machine learning applications today are more focused on things like computer vision or natural language processing or speech processing. And molecular machine learning is basically what it sounds. Instead of having, vision or speech or text as input, you instead have molecules. The field of chemoinformatics has created a number of standard file representations, a number of standard molecular formats, which can be fed into molecular machine learning systems to make predictions about the properties of molecules. Um, Now, the thing about molecular machine learning is that I think there's a lot of challenges that are unique to molecular machine learning that are not as apparent in other subfields of machine learning. Um, for one, I think the biggest challenge, I'd argue perhaps the most fundamental challenge, is that data is perennially scarce in molecular machine learning. For a lot of machine learning applications, let's say you're running perhaps a recommender system for an e-commerce website. There's a pretty standard pipeline, which can still be very challenging for gathering new data. You can have recommenders kind of manually walk through and annotate new data points. However, in molecular machine learning, the creation of a new data point is an experimental endeavor, which can require the input of a highly trained scientist or technician to perform, and is often very expensive. The cost of reagents, the cost of material, the cost of running a clean experimental lab mean that access to data is a critical resource. In general, companies are also much more uh, thoughtful about when to open source data. The data sets of a pharmaceutical company often contain its core proprietary knowledge, and more often than not, it's very hard to get access to data outside certain kind of free resources, some of which DeepChem helps maintain, that provide access to governmentally gathered or academically gathered data sets. But in general, when doing molecular machine learning, you have to plan for data scarcity as a fact rather than hoping that there's a larger data sets will become available, which I think they slowly will be, but it will be a much slower pipeline than it is for the rest of machine learning. The other challenge is that molecules come in all sorts of shapes and sizes. There's also a lot of different things we want to do with molecules. This means that there are a variety of different featurization techniques that crop up in molecular machine learning. This is a phenomenon that is, I think, less common elsewhere in machine learning. If you're, say, dealing with text, oftentimes perhaps you just one-hot encode it. Or if you're dealing with images, oftentimes you can just turn it into an array in like an RGB format in a standard way. For molecules, there's not one canonical encoding that most people use. Instead, there's something like an entire array. I believe DeepChem supports something like 20 or 30 different featurization techniques, with more being added every day. So there's a very broad range of different properties. And the reason this happens is that molecules have all sorts of properties. Some of them are quantum mechanical, some of them are electrical, some of them are physical, some of them are, you could even say, biological. So depending on the application at hand, you have to use a different uh, feature representation for the molecule. Now, there's been, I think, some work trying to move towards something like a universal featurization, but for now, this is still like a wild pipe dream. And that's more of a open research question than something that can happen in practice. In practice, a good production pipeline should have multiple featurizers that it can use for different tasks. And just to kind of provide a little bit more color to the previous slide, here's a few examples of some of the featurizers that DeepChem supports. The ECFP fingerprints you could think of as akin to bag of words in a natural language processing setup. A Coulomb matrix is a representation of the electrical behavior of a molecule. You can think of the ijth index as tracking the Coulomb force between two atoms, I and J. You can also have things like graph convolutional featurizations. These featurizations annotate each atom with a local descriptor, which are then combined through various deep convolutional networks. There are also things like grid featurizations, where a known three-dimensional information about stable configurations of molecules are used as input to the machine learning system. So in general, being able to create a production molecular machine learning pipeline means that you should have a working knowledge of the different types of featurizers that are available and understand when each type of featurization is relevant or not relevant. The other, and I would argue perhaps biggest challenge facing molecular machine learning is that it is more akin to a field of science than it is to a field of engineering, in that we need our systems to be able to make predictions on molecules that have never been seen before. And the reason for this is simple. If you're a company that's making a new therapeutic, you want to own that molecule. You want to make a new molecule no one else has ever made, otherwise you won't get patent protection. This means that your molecular machine learning pipelines must be capable of generalizing to molecules that are far from the source training data. This can make it quite challenging for a molecular machine learning pipeline to yield fruitful results. Later in this talk, I'll say a few words about ways you can test and improve the generalization, but the thing to beware of here is that a model which, say, performs very adequately for an e-commerce setting may lack predictive power uh, when translated to something like a molecular machine learning setting. You need to be much more rigorous and much more tough on your models. And you can't just let them slide with being able to understand the training data. They have to generalize in order to be useful. So part of one of the core resources that DeepChem maintains for practitioners is something called the Molecule Net Benchmark Suite. This is a collection of some, in the original release, V1 release a few years ago, 17 data sets. And in our run-up to the upcoming V2 release, probably end of this year or later, we have something like 30 plus data sets. It's a broad collection of different data sets for different use cases of molecular machine learning. And as I mentioned earlier, there's an entire array of things that you want to do with molecules. They can range from the quantum mechanical up to the physiological. And you often have to choose the right training data, the right featurization, and the right model in order to be able to meaningfully make predictions across this broad range of potential applications. So with that, I think I will kind of shift a bit into talking a bit about tips and traps. As you begin to build molecular machine learning pipelines using, say, models that are implemented in DeepChem or elsewhere, there's a number of pitfalls that you can fall into. while you're building these systems. I think the first pitfall, coming back to our earlier point about generalization, is that the choice of split is critical. Now, you might be asking, what is a split here? What does it mean? And the key point here is that when you are training a model, you oftentimes separate the data set at hand into training, validation, or test. And there's a question of how do you do the separation? Oftentimes in machine learning, you do something like a random split. You say that, okay, I will randomly subsample, say 70% of the data for training, I'll use 20% for validation, and a final 10% for a holdout testing set. This, unfortunately, is unlikely to yield good results when doing molecular machine learning. The reason for this is that your model is likely going to, on this retrospective random split, look like it has very strong predictive accuracy. And the reason for this is that models have trouble generalizing the further away you get from the training data. So if you do a random split, it's very likely you'll have a close neighbor of every molecule you want to make a prediction on in the training set. This means that the model faces an artificially easy benchmark. So if you want to have a better sense of the actual predictive performance in these models, you oftentimes need to do something like a scaffold split. This is a split that uses the chemical structure of molecules to separate into training, validation, and split. And DeepChem maintains a broad variety of different splitters, as we call them, different ways of separating a data set into training, validation, and test. So if you're creating a molecular machine learning pipeline, be very thoughtful about how you can ensure that your training set is adequately separated from your validation set. Otherwise, when you actually test the model out live, you're going to face some unpleasant realities that your 90% accuracy model gets 10% accuracy in live performance. So this is something to watch out for, and this is a trap that will definitely befall even experienced practitioners, because it's very hard to tell how well a model generalizes. And the only real way is to test it prospectively over time, so you get a real sense for true future performance by testing it thoroughly over multiple experimental cycles. And I think that, just to drive that point in even further, I think that one of the biggest challenges that you'll face here is that distribution shift is also to be expected. So if you are, say, working with a pharmaceutical company, oftentimes they might at present be working on a particular series of molecules that they're very excited about. But then all of a sudden they might have a breakthrough on the chemical side and shift their focus to an entirely different family of molecules. This means that because of the shift in distribution, your model trained on the previous set of molecules might entirely fail to generalize to a new set of molecules. So again, when you're thinking through production molecular machine learning pipelines, you have to think very carefully about, did I do the split correctly? But even more so than that, has my underlying distribution shifted because I'm in a new region of chemical space? In many ways, I think this is a challenge that you do run into in, say, e-commerce applications as well. There is distribution shift where customers have new preferences. The challenge, I'd say, that's new to molecular machine learning is that the distribution shift can be very rapid, in that, not very rapid, I mean very abrupt in the chemical space, which means that the set of molecules that can be created by a new set of experiments might have absolutely nothing to do with the set of molecules that were created in the earlier training data. So it's entirely possible that you have a molecular machine learning pipeline whose predictive power drops to zero or random when there's a distribution shift. So this is something to be aware of, and also to educate any counterparts you have that are working chemists, and to create expectations that models will at times fail to generalize the further away you get from known historical data. The other thing I'd say that is that reproducing research results can be very hard. This is true throughout machine learning. We know that a result that's in a research paper can be quite difficult to reproduce in practice, especially if you don't have the team that produced the original results on dial. I think there's a few extra challenges in the field of molecular machine learning. For one, I think you need to have a pretty decent working knowledge of chemistry in order to check whether some of the representations of molecules were handled correctly. There's a number of subtle points with things like, did you canonicalize the representation of the molecule, which can end up yielding big results. There's other things like hydrogenation, like molecules, it turns out, in a solution, they might have different numbers of hydrogen uh, H plus ions attached to them. And depending on the pH in which you're considering the molecule, this can make a difference. There's also subtle points like chirality, and that is like there are mirror representations of a molecule. And having information about the chiral structure can sometimes lead to a big difference in practical performance. You also need to worry about all the usual machine learning things like details about training, details about data. I think another issue that we found when we first created the molecule net benchmark suite, we chose suggested experimental guidelines that matched the expected underlying distribution, but were in some cases hard to reproduce for new practitioners. This unfortunately has meant that some of the benchmark results you'll see on MoleculeNet papers are hard to reproduce because they depend on randomness that the new paper author has used. This is something we're looking to fix in our upcoming MoleculeNet V2 release, but for now, take benchmark results with large grains of salt and expect to have severe difficulties when reproducing papers in production settings. And I think I already hit a good number of these points, but in general, domain knowledge is critically useful. If you're building a production machine learning pipeline, it's really, I think, necessary to have a close rapport with working chemists and biologists and other scientists who can help scrutinize the scientific validity of the model that's created. And if, whenever possible, work closely with experimental groups that can actually test whether your model makes sense by running live experiments. The only real test of a molecular machine learning system is its ability to predict future behavior. So if you have close reports with teams that can help you do this testing as part of the model development, that will considerably increase the actual performance of your system and make a real difference. Don't only trust retrospective numbers. Insist on the ability to get prospective numbers when building these pipelines. So with that, as I'm kind of beginning to wrap up, I'll say a few words about more resources to get involved with the field of molecular machine learning. This is a subfield that's really exploded, I think, over the last 5 years. There's just so many applications, as we've seen with COVID, where the ability to design therapeutics more rapidly can make a real difference for people. There's also adjacent fields like agriculture, like material science, materials design, where many of these techniques carry over and can have real impact on real world problems. I think kind of the first place I'd recommend is, sign up for the DeepChem forums. There's a pretty active community of people who are on there discussing various models, various, techniques, issues they've run into in practice. We also post our regular DeepChem developer minutes on the forums so you can see what our development team is up to. You're also, of course, welcome to join in. It's an open community, so anyone who's interested can dial into the developer calls for DeepChem. We also have a live chat room. There's usually a good number of people hanging out in there. It's a good place to ask questions if there's some subtle point that you're running into. We have a book. It's called Deep Learning for the Life Sciences. Discusses broadly the use of deep learning for life science applications, and all of it is kind of implemented and maintained as part of the DeepChem broader framework and community. And I think with that, I'll just wrap up by saying that, in general, as a community, we're still walking that long path from research to production grade. We really do need help from experienced machine learning practitioners like yourselves who have experience in other domains and really building these production machine learning systems. We're an open source community, kind of welcome new practitioners, new contributors. So would love to have more folks involved. And I think just speaking for myself, I think this is an incredibly enriching area of machine learning where, there's not really concerns about privacy or kind of some of the issues that we face elsewhere, but really rather hard scientific challenges that can make a real impact for people trying to discover new medicine or design new materials and do lots of cool things. Okay, with that, I think that's about everything I had formally prepared for a talk, and I will punt over for questions.
Thank you, Bharath. So there's a couple of questions on the Q&A. There's actually 3. So as I speak, they're being added. So maybe you can take them in turn. And since we're in the Q&A session, you can probably answer them live. I think that would be best.
Okay. 1 second, I'm just trying to pull up my... Sorry, I might have to stop screen sharing if that's all right. Having trouble.
Yeah.
Pulling.
That should be fine.
Ah, there. Okay, so first question from Sergi Sparks. My apologies if I'm mispronouncing that. Can your simulations help us model drug effectiveness with machine learning and stop killing animals with drug overdosing? It's a really good question. I think the unfortunate answer is probably not. The reason for this is that biology is immensely complicated. We have a very crude understanding of the systems that deal with toxicity in live kind of animals or humans. At best, we have very crude models that capture a portion of this. But if you talk to any kind of working drug discovery scientists, they'll often have a story for a molecule that worked well in every non-animal test and machine learning model. But at the very end, you gave it to, say, rats in a test, and they all died instantaneously. So it's a cruel practice. It's one I don't sit easily with. But the alternative is testing on human volunteers. And I would much rather a rat die than a human die. So I think this is one of the unfortunate situations where, due to our lack of scientific knowledge, I don't think machine learning is at a place where we can really get rid of animal trials. It might be possible to be able to do fewer animal trials, especially as we continue building up open data sets about toxicity. So I think there is a long path to lessen the need slowly on animal overdosing, but I expect that for the foreseeable future, we will need to test on animals in order to avoid human deaths when creating new therapeutics. Okay, with that, we'll move to the next question from Giannis. You touched about the role of embedding environment of molecules, for example, immersed in water or not. Is there a further complexity introduced by environmental parameters such as temperature, pressure, extracellular matrix, etc.? This is a great question. And the answer is yes, absolutely. Like there's a lot of details that crop up in practice, like for example, pH I mentioned briefly. There's also things like, do you have other ions in the environment? In materials, like pressure, I think is really critical. Like a lot of superconducting materials only superconduct at very high pressures, like gigapascals and things like that. So oftentimes what we end up having to do is that we can't model the full range of environmental parameters exactly. So we try to pick a relevant subset of them for the problem at hand. If you are, say, dealing with the problem of solubility in water, oftentimes you do need to care about things like the pH. You can usually assume that pressure is standard unless you have an exotic environment, so you can ignore that in your data. So I think there's no extracellular matrix in that case, for example. So it really is a domain-specific modeling problem that you have to just think about the physical system at hand and the factors that are relevant. Unfortunately, a lot of this field is still deeply tied in with the scientific underpinnings. So there's no easy way around just thinking about like the domain system at hand. Okay, question 3 by Kumar. Can GANs be used in this domain? Yeah, absolutely. I think there's a very thriving field of GANs for molecular design, where you use a GAN essentially to sample new molecules. More broadly, there's a very thriving field of just generative machine learning for molecules, things like variational autoencoders for molecules, things like there's increasing interest in things like language models applied to molecules encoded as SMILES strings, which are a representation of a molecule basically as a word in a standard format that is convertible back and forth between a molecular graph. So I think there's a lot of scope for generative applications and GANs. And I bet there's hundreds of papers that people have been writing about this. So yes, absolutely. One more general question. How do you think intellectual property, but also work compensation should take place in an open source project? Are you handicapped compared to proprietary research firms, which can lure abundantly more funding? I think the answers to this are unfortunately the same as they are for every other open source project, in that open source projects really are just driven by the passion of people who have the wherewithal to work on it. So speaking for myself, like I'm able to do this because I use DeepChem in say other engagements for in consulting roles. But for off peak hours when I'm not working on other things, I have the ability to continue working on developing DeepChem. This really does, we have other contributors who are scientists at various institutions or graduate students or developers who are contributing in their evenings and night times. I think, yes, there is a definite handicap when compared with a very well-funded proprietary research firm that can create a large closed source package. But I think the flip side is that DeepChem as a project has been around over 5 years now. In that time, I've known a number of companies that have actually uh, grown big and also closed down. Like it's hard, especially at like a startup or like sometimes in a corporate environment, to nurture software on the long time scales needed to create really stable systems, which I think is the same argument for something like Linux, especially if you're creating software of complexity. Having an open source community that supports it across multiple jobs can be a great strength, in addition to being a weakness. That said, if you want to help us, like, and we are looking for donors, we have some AWS usage for maintaining our data sets where we need a little bit of funding. We also could use more developers. So yes, there is a handicap. We try the best we can to work around it, but it's definitely a challenging field. A question from Catherine. What are the kinds of targets you define for this machine learning for drug discoveries? That's a great question and one I didn't go into. So a target is kind of a term used commonly in the biotech and pharmaceutical industry for usually a protein that serves some biological function in the body. So when designing molecules for therapeutics, we often have to take a reductive stance. Diseases are complicated. There are many, many factors, but we try to winnow this down to one hypothesis about the core driver. For example, perhaps there's a mutation in one particular genome that causes a downstream protein encoded by that genome to misfold. Um, so then you might use that taking out that protein is your target. You might say have a goal to uh, cause the protein to be degraded. There are ways to recruit the body's cleanup engine, so to speak, by adding a signal to that protein that it's malformed, which makes it be cleaned up by the body's kind of natural cycling mechanisms. There are other times where you have a protein that's overexpressed, and maybe you want to shut down part of it so that you lower the expression level to this normal realm. There are other times where, sometimes in things like cancer, where you want to basically turn off a protein entirely if that's a protein that's used only by cancer cells. This turns out to be extraordinarily challenging, but a few drugs, notably I think Gleevec, took this route where they shut down, I believe, the protein c-Abl. So really a target here is a protein or related biological molecule that you hypothesize is the root driver of the disease. Okay, another question from Catherine. Do you think quantum computing will govern this kind of machine learning field and will open the opportunity to create new models over this technology? That's a great question. I think there's one in which there's still some scientific openness in terms of like the answer. It's still hard to say to what degree quantum effects really play a role in drug design. I think there's good evidence that it's really more electrical behavior and physical chemistry that play more of a role than quantum effects. But there are known edge cases where there are molecules where quantum behavior is suspected to be important. Going a little further afield, photosynthesis is an entirely quantum phenomena that has some strange behavior attached with it. So if we know in biology sometimes quantum mechanics is really important, we also know in other cases where it doesn't seem to be the core driver. So I think for now it's a open question, like how deep the role of quantum mechanics goes in drug discovery. And if in places where quantum mechanics is critical, I think quantum computers will have like a big impact. In places where it's really not the core driver, I don't know. We don't know what quantum computers can do yet, so we'll have to wait and see. I think question, what are the most significant machine learning success stories in this area of molecular biology directly impacting humanity? That is a great question. I think it's a hard one to answer because I don't know if I can point at any one medicine that has been discovered by machine learning yet. The field of this field just tends to move slowly. With before COVID in general, you'd think 15 to 20 years to bring a drug from conception to human patients. For example, like gene editing became first viable, I believe, in like the early 90s or thereabouts. But it's only very recently that the first commercial gene editing therapeutic was approved for use in patients. And the reason is there's a lot of complexity that just pops up. So there's been a variety of startups working in this field, I think, since 2012, 2013, after that. But I don't believe anyone has gotten so far as to have a molecule that was machine learning enabled make it to the clinic. There are earlier versions of this technology which use simpler physics-inspired computational techniques that I believe have made an impact. But again, it's hard to prove that. Like if you used MATLAB while you're designing, say, a new material or car, can you call that a MATLAB-driven car? Kind of, sort of. There was a pretty big contingent of human work in there. I think it's going to be the same in this field. So I think with that, that's about all the questions out there. Any other questions?
Yeah, I think that was it. Thank you so much, Bharath. I had one quick final question. You mentioned that in the area of drift, for example, that the changes in this space can be abrupt. I don't think I quite understood why the abruptness happens. Could you elaborate?
Yes, absolutely. And I think that was a bad word choice on my part. So I think what I meant by that is that chemical space is very vast. There's something like 10 to the 60 molecules that are believed to be potentially drug-like. And there's a much broader field of adjacent molecules. So oftentimes when you are creating a model, the drop-off in performance can be rapid as you walk away from it in chemical space. So if you have molecules that are, yep.
Okay.
So the unfortunate thing is that unlike Newton's laws, we don't have these powerful general purpose tools. Instead, we have, I don't know, Newton's local town council park memorandum of understanding.
Okay. Yeah, right. So I think just to make sure I understood, what you're kind of saying is the space is vast and we don't have good navigation tools. So if we step out of line, things could change
Exactly.
very quickly.
Like, let's say that your experimental team comes up with an amazing new technique to make an entirely new family of molecules. You as a molecular machine learning practitioner would be like, oh shit, it's all going to break. Which I think, but be skeptical of your models, I think is
Interesting. Good to know. So thank you so much. So we have 1 minute left. If anybody has any questions, please do post them on the Q&A. So we'll wait a minute and see if that happens.